We now turn our attention to the cell membrane, a hydrophobic structure in an aqueous world. It forms the border of the cell between the cytoplasm and the environment. Shown here on your left is a molecular model of what the membrane actually looks like, and you can see all the phospholipids. An important concept to understand before talking about membranes is that chemical structure determines functional properties. A great example is how the amount of saturation and the size of lipids determines their liquefaction temperature. Unsaturated lipids, those with double bonds such as safflower oil, are liquid at room temperature. The double bonds cause a kink in the molecule's chemical structure, making it more difficult to pack into a solid and requiring a lower temperature for it to do that. Butter is about the same size as safflower oil, but it does not contain carbon double bonds. It can pack together more efficiently. And will solidify at a higher temperature. Thus, it is a soft solid at room temperature. Beeswax is made from a lipid that is linear and much longer than butter. At room temperature, it forms a stronger solid. These properties are important for membranes, as you will see in a minute. Lipids constitute 70% of the membrane, and these lipids contain glycerol and fatty acids. The fatty acids contain a COOH group, which is called a carboxyl group, and that is then connected to long alkyl chains. The alkyl chain may have a double bond in it, or two. Two fatty acids are connected to each glycerol, with most of the lipids in a bacterial cell membrane being phospholipids. The third hydroxyl in the glycerol molecule connects to a phosphate that contains a hydrophilic or water-loving molecule. In the example here, it's ethanolamine. Lipids thus have parts that are hydrophobic, hate water, which are the alkyl chains, and hydrophilic, that love water. Membranes also contain hapenoids, which are similar in structure to sterols in eukaryotic membranes. They add stability to the membrane. The phospholipids will orient in a double-sided surface, with the hydrophilic heads facing outward towards water and the interior of the membrane containing the hydrophobic fatty acid tails. This arrangement is called a lipid bilayer. Phospholipids are free to move laterally on the surface, as you can see in this upper right figure. Proteins decorate the membrane and make up about 30% of the composition. The term fluid mosaic describes the fluidity of the membrane, the lipids, and its decoration with proteins, the mosaic. At a given temperature, a lipid bilayer with no unsaturated fatty acids will have a lower permeability and be less fluid than a membrane with higher saturated fatty acids. At a given temperature, a lipid bilayer with no unsaturated fatty acids will have a lower permeability and be less fluid. As you increase the number of unsaturated lipids in a membrane, it becomes more fluid and more permeable. The fluidity of the membrane is also reduced by the hapenoid concentration. Membrane fluidity increases as the temperature increases. Given all this, consider the following question. Microbial survival depends on membrane lipid homeostasis and requires the ability to adjust the lipid concentration to Microbial survival depends on membrane lipid homeostasis and requires the ability to adjust lipid composition to acclimate the cell to different environments. A hypothetical bacterium Bacterium anonymous grows normally at 25 degrees centigrade and has a membrane lipid content of 50% saturated fatty acids and 50% unsaturated fatty acids. Remember, saturated fatty acids are more solid and unsaturated fatty acids are more liquid at a given temperature. What would you expect the membrane lipid composition of B anonymous to be if the bacterium was shifted to a lower growth temperature of 10 degrees centigrade? The correct answer is A. You would expect an increase in unsaturated fatty acids. As the temperature cools, the membrane would become less permeable and less fluid. To counteract this, B anonymous would take out saturated fatty acids and add in unsaturated fatty acids. Membrane permeability is not the same for every molecule. The membrane is what's called a semi-permeable barrier. The scale here shown on this slide on the left shows the rate at which various particles can penetrate a membrane. The higher the number, the higher the permeability. Gases such as oxygen, carbon dioxide, and nitrogen can pass easily through a membrane and are not deterred. Water, a small uncharged but polar molecule, can still pass through at a reasonable rate. As a compound gets larger, for example glucose or sucrose, it has greater difficulty getting through. Finally, the least permeable compounds are charged molecules such as ions. Things like glucose, sucrose, and ions will need gates to pass through to get through the membrane at any kind of reasonable rate. The cell needs to get some of these molecules across the membrane. How is this accomplished? For gases, they simply pass through the membrane in a process called passive diffusion. Sometimes passive diffusion is too slow, as is the case for water. If you don't need to concentrate the substance, a cell uses facilitated diffusion. In the case of water, a protein called aquaporin, see the image at right, provides a gate for the water to pass through. Figure 314 of Through the Microscope shows an animation of facilitated diffusion. As you can see, we start at the beginning with all the 100 molecules on the inside. As the gate allows molecules to get through, they distribute. At this point right now, you see that there are 31 molecules on the inside, or 37, and it keeps going up, and about 59 on the inside. If we let this run long enough, there would be a near equal number of molecules on the inside as the outside. In other words, facilitated diffusion does not concentrate a molecule against a gradient. If a cell wants to concentrate a compound on the inside, it must use energy to do this. In these cases, a protein binds the molecule, then some form of energy will be used to move the molecule inside. This energy can be a gradient, such as the protons accumulated on the outside of the membrane, or ATP. Active transporters are molecular pumps. One example of an active transporter is porter systems. A uniporter can bind the target and move it across the membrane with no compound involved. A symporter will transport the target and will use a second molecule to drive the transport. In these cases, the second molecule is at a higher concentration on the outside and can drive the transport because it falls down its gradient. Often this second molecule is a hydrogen ion, but it can be other things such as sodium. An example of a symporter is the branched chain amino acid transporter. It moves leucine, isoleucine, and valine into the cell at the cost of also transporting hydrogen ions into the cell. In an antiporter, the second molecule is inside and at a higher concentration. The protein moves the second molecule to the outside with the movement link to the target's transport to the inside. Another type of active transport is ATP binding cassette or ABC transporters. There is a solute binding protein that binds the target. It then attaches to the transport channel. Finally, a conformational change is driven by the hydrolysis of ATP by the ATP binding protein. This change causes a gate to open and the target moves to the inside of the cell. Figure 316 demonstrates this process. Here you see the maltose binding protein. Here is maltose, the thing to be transported. ATP is a little yellow circle. Here's the maltose transporter. So we'll click it to begin, and you can see the maltose binding protein binds to ATP bind in the bottom. There's a conformational change and it transports across. We'll wait one more time. Maltose binding protein binds. There's 2 ATP there. It transports across. And finally, once more, now 2 ATP bind, and the maltose binding protein transports across. Finally, a third method of active transport is group translocation. Example of this is the phosphotransferase system of E. coli. PTS first charges itself with phosphate from phosphoenolpyruvate. It then binds glucose and modifies it as it passes into the cell. You can see here's phosphoenolpyruvate, a phosphate comes, glucose then binds and is transported. Phosphate is split off PEP, the glucose binds, and it's transported. You can see it changes as it goes across the membrane. We wait for another glucose to bind, it binds, and it comes across the membrane. Once more, glucose gets transported. And you can see now glucose is accumulating on the inside of the cell against the concentration gradient. Let's see if you have a good grasp of these transport systems. In bacteria, all transport systems are driven by... The correct answer is C, gradients. All of these systems depend on gradients. Passive diffusion and facilitated diffusion depend on the gradient of the target for the target to be transported into the cell. Porters depend on the gradient of the second molecule that helps drive the transport. Finally, group translocation and ABC transporters depend on high-energy phosphate compounds such as phosphoenolpyruvate and ATP. The cell creates high-energy phosphates, most often using a gradient, as you will see in the metabolism chapter. As a summary exercise, I encourage you to fill out this table on various transport systems that we just covered. This is a great active study tool to help you understand how these systems work and to better remember them. After you have made your attempt, check out table 3.1 of Through the Microscope for the answers. Transport is not just a matter of getting things inside the cell. Cells will often want to get molecules outside the cell. For some small waste products, they may be moved out by antiporters. In other cases, efflux systems will transport unwanted small molecules outside the cell. Bacteria also need to transport proteins across the membrane, either to insert them in the cell wall or to send them into the environment. Microorganisms secrete proteins using secretion complexes, and there are 7 known systems. Efflux pumps move molecules out of the cell, and of course, there are different types in various bacteria. These pumps tend to be non-specific in what they move out. They will transport things like antibiotics, heavy metals, organic pollutants, plant-produced compounds, quorum-sensing signals, and bacterial metabolites. The figure shows the configuration of known systems. Efflux pumps can be one way to become resistant to antibiotics. The bacterium simply makes more of the efflux pump, which pumps all the antibiotic out of the cell and keeps it at a concentration low enough that it doesn't inhibit or kill the microorganism. Several secretion systems are known depending upon the bacteria. I want you to know that these systems are necessary for the transport of proteins out of the cytoplasm. TAT and SEX systems are important for protein transport in gram-positive cells. There are also type 1 to type 7 secretion systems in gram-negative cells. we will talk more about secretion systems in the module on infectious disease. For right now, I want you to know that they exist, that TAT and SEC are used in gram-positive bacteria, and that there are type 1 to type 7 systems also in gram-negative bacteria. In summary, membranes serve three general purposes. They create a selective permeability barrier, they serve as an anchor for membrane proteins involved in transport, structure, and energy generation, and they help to generate and conserve energy. That's it for this section. In the next section, we'll look at the outer layers.